we're kind of getting to the end here in terms of going through the organelles. I mean, we still have to talk about the plasma membrane. That's a big organelle, real important organelle. But we're making our way through, especially the components of the endomembrane system. We've kind of done that. We're leaving out chloroplasts. So this is one of the final organelles that we're going to talk about. And peroxisomes are really unique because, think about their role in detoxifying the cell, in kind of regulating catabolism of lipids. Cells have many peroxisomes, anywhere from 100 to 1,000 peroxisomes, depending not only on the type of cell that it is, but also the state of the cell, right? So if you just drank a bottle of Jack Daniels, your peroxisome count in your liver is going to shoot way up, right? To detoxify the environment of the cells. And that's, primarily its role in the cell is regulation of catabolism and also detoxification. Right. So unlike the ER, let me make sure this audio is working. It's working. Okay. So unlike the ER, or like vesicles that come out of the ER directly, so proteins can be brought into the peroxisome that are already completely folded, right? So most of the examples that we've looked at regarding import in the endoplasmic reticulum or the mitochondria involve the use of chaperones, right? So the chaperone has to bind to the protein to keep it in a linear fashion so that it can move into the translocon, nuclear pore complex, the Tom complex, whatever. Peroxisomes, the thing can stay folded up and get inside. And that's part of like the rapid response nature that peroxisomes have. Right? Most of the proteins synthesize on free ribosomes, right? Those are going to be your enzymatic proteins, metabolizing proteins. There are some that come from the ER, but those are typically the transmembrane proteins of the peroxisomes. All right, so this is peroxisome. Sometimes you get like these dense regions inside the organelle that are usually like these crystalline structures, right? So I think this is uric acid or something that's building up in there, and they just start to densify and crystallize. So important roles in fatty acid oxidation, right? So we've talked about one form of oxidation already that involves these long chain lipids. What was that form that we talked about? Beta oxidation, right? So beta oxidation is a very efficient form of breaking down long chain fatty acids. They produce shorter chains and produce ultimately a lot of molecules of ATP from that. One of the byproducts of that is toxic hydrogen peroxide. So this is a byproduct of that fatty chain breakdown. And hydrogen peroxide is highly oxidizing, very dangerous for the cell. Oxidizing agents can damage DNA, damage proteins, and you want to keep that in balance while also maintaining enough energy for the cell. Right? So there's always going to be a little waste, but you don't want that to get too high, and you're always going to need to get energy, so you're always going to need to make some waste. And the peroxisome's role is really kind of balancing that catabolism with detoxification. Right, so there's your reaction that's going on inside of peroxisomes, the beta oxidation, and it's providing acetyl-CoA. It's ultimately going to go to the mitochondria. So if you look at that chemical reaction up there, what are you doing here? So that's an oxidation reaction. You're producing a double bond between CH2 and CH2. Right, so you're shortening that molecule, and that's what beta oxidation is. It's shortening the polymer. It creates peroxide as a product, and the chief enzyme in degrading hydrogen peroxide is catalase. You probably heard of that before. Didn't we do a lab on that? Did a lab peroxidase, right? All right, catalase will break down hydrogen peroxide, which is a further oxidation reaction, right, into water and oxygen, or it can break it down into another organic like with another organic compound. All right, so peroxisomes, they are an important site of lipid synthesis as well. So plasmalogens, these are very prevalent in the axons of neurons, right? So plasmalogens make up the myelin sheath that insulates the neuron. Very important structure in nerve conduction. Who's taken A&P and studying that? Right, the voltage gated channels that produce that ion exchange. Without that myelin, that integrity, you can't maintain the voltage gradient. Okay, so how do peroxisomes get assembled? Well, they can actually get derived two different ways. One is they can come from previous peroxisomes that just basically split. Another way is that they can derive directly from the endoplasmic reticulum, so they can kind of bud off as these vesicles. And the third way is that they can you can fuse two peroxisomes together to produce a new larger peroxisome. All right, so one set of proteins that kind of directs the fusion of these peroxisomes are these V1 and V2 proteins that express in the endoplasmic reticulum as a transmembrane protein. So with the peroxisome, you have like an internal region, which we call the matrix, just like the mitochondrial internal region is called the matrix. And proteins that are going to go there, like I said, are usually synthesized on free ribosomes, but membrane proteins of the peroxisome are going to be derived from the endoplasmic reticulum. What happens with these V1 and V2 molecules is they merge together on the membrane of the peroxisome and they produce what's called an importomer. The importomer is just a big channel that folded proteins that were synthesized on free ribosomes can translocate through and then now derive or exist in the matrix of the peroxisome. Right, so there you got your peroxisomal endoplasmic reticulum budding off. And one of them has the V1 protein set, the other one has the V2 protein set. They fuse together and it kind of helps assemble this importomer channel. The importomer channel is what can receive cytosolic free ribosomal proteins that were translated there. So no signal sequence involved in this sort of import. All right, however, some imported proteins involve a peroximal targeting signal, or PTS1. Right, this is a signal that's found at the carboxy terminus, and there is a shuttle protein that is in high abundance in the cytoplasm called PEX5. And PEX5 will go out and it'll bind to the PTS1 signal sequence, and then it'll deliver it to the peroxisome for import. So when PEX5 binds its cargo, right, it comes to the peroxisome, it binds to this membrane receptor complex. It's made up of a bunch of other proteins. You don't need to memorize those. They're just other PEX proteins. And you get this pore that allows these specific proteins that have this PTS1 signaling sequence into the peroxisome. Okay, so these proteins can also come, or do also come, from free ribosomes that are in the cytosol. right? But they have a signal sequence, right? The ones that come through the importer do not have a signal sequence. Right, so those are your cytosolic ribosomes producing cytosolic proteins that are directed to the peroxisome because they contain a signal sequence, PTS1. So PEX5 is a protein that shuttles it, right? So it's a protein that's moving through in the cell through diffusion, and it will pick up these proteins and deliver them to the peroxisome through diffusion. And it will dock there at these series of other proteins, and then it will allow the translocation of it into the peroxisome. Okay, so like I said before, how new peroxisomes can form, they can form de novo, right? So within. So these form from the endoplasmic reticulum, right? So vesicles budding out. They can also fuse together and they can also divide. So this, having these things be able to derive from 3 different ways is what allows them to really rapidly regenerate in response to some sort of cellular cue, like increased toxins or increased need to break down fatty acids. All right, so there we've got 2 new peroxisomes budding off of the ER, and they're fusing together. They're creating that importer. They can grow and get bigger, and they can also divide, produce multiple peroxisomes. And those new peroxisomes can grow and divide almost just like cell division occurs. Right? Not too bad, right? Okay. Now this chapter 13, is pretty massive. We're probably gonna omit the next section, which is on myosin motors. You guys usually study skeletal movement pretty well in Bio 1, and then you get it again in A&P. And usually they get pretty nitty-gritty with it. But if you take my molecular biology class, you have to take that. We'll get into the myosin motors a lot more. So 13.2 will probably get omitted, but we will, following up on after the exam, we'll get into 13.3 and 13.4, which get into microtubules and also into kinesins and other motors. But the first section of the chapter has to do with one of the most basic scaffold proteins in the cell, which is actin. Actin is the most abundant and really important cytoskeletal protein in the cell. It's responsible for movement, shape, transport, all these different things. All right, so where does it fall in terms of all the different filaments? Well, there's 3 types. There's actin, which we're going to talk about in this chapter, also called microfilaments. There are also microtubules. What's the main role of microtubules? They're involved in cell division. Microtubules are cell division. And then intermediate filaments, right? So these are filaments that are kind of between the small size of actin and the larger microtubule proteins. Right? That's why actin filaments are called microfilaments, because they're very abundant in the cell. Actin protein makes up one of the more substantial proteins in most cell types. But their size, in terms of their thickness and whatnot, exists somewhere between, is the smallest of the 3. All right, so how is it built? We're going to get into the structure of actin in some significant detail. At the most basic, like monomer level, it is a globular protein called G-actin. That's what the G stands for, globular. And these monomers come together ultimately to form filamentous actin, which is F-actin. So these G proteins start to form together in multiple orientations. They come together as dimers, trimers, and when that happens, it acts as like a like a bud for growth, right? So think about like if you're growing a crystal, you just need like a little speck of the shape and the right environment, and the crystal will grow. And that's what these nuclei are for actin. Once you get, the formation of some dimers and trimers, we call that an actin nucleus, the thing can just take off, right? And start polymerizing. So actin does have polarity. It's got a plus end and a minus end, and these ends are very important in its development and polymerization. Overall, the plus end grows a lot faster than the minus end. See a little more detail about that. So these globular subunits, they assemble, like they kind of stick onto the growing polymerizing molecule in a certain orientation, but they form like a right-handed helical filament. Okay. So what other molecule that we have studied is a right-handed helical shape? Yeah, one of the DNAs, DNA A, right? That's the one that was discovered by Watson and Crick. All right. DNA B is also right helical, and DNA Z is left. Okay, so and what's forming this helix is like the orientation of the G actins and how they stick on, and that angle that they do that at. So every time a actin monomer gets onto the growing chain, that requires an ATP. So every single monomer that comes onto the growing chain requires ATP, and every single monomer that comes off the chain, you're losing ADP from that. Let's look at some figures here. So there's the G actin. And you can see the minus and the plus end, and that's going to kind of configure in the final filamentous form, like how it's growing and how you get the minus end and the plus end. You can see it growing into dimers and then trimers. And at that point, you have what's called a nucleus so that the polymerization can take off. And you can see how it's like forming that helical structure to make that final filament. All right, ATP use and the availability of ATP in the cell is very important in actin dynamics. So when you have these monomers, right, these globular proteins, imagine, there is ATP conjugated to it. And the ones that are the globular proteins that are going to the plus end, right, have the ATP. The ones that are at the minus end have ADP on it. And the plus end is growing more rapidly than the minus end is depolymerizing. Okay, this is like a steady state of the actin molecule that's keeping it, at a particular length for a while, but then it can take off and grow. And they call this treadmilling, right? So it's like a turnover of the filament where you're having these ATP monomers get rapidly added to the plus end, and then in general, they're more slowly depolymerizing at the minus end, and it's keeping actin in its steady state. I want to show you actually a video of this online. I just have to find it, but I think I'll be able to find it. That's it. There's two I want to show you. They're really quick. I don't need any sound on here. God. So you can see these things, how they're, all right, now this, what end would this be, plus or minus? Plus. It's not showing you it doesn't show you the minus end. And the other one I want to see, this one may be harder to find. That one. Yeah, I don't know. I don't see it. Okay. Well, it shows like all the actin polymerization that's going on in a cell and how it's very rapid. If I find it later, I'll post it on Canvas for you guys. Okay, so this is treadmilling, right? So it's the constant dynamic interchange of this ATP and ADP. But just appreciate, how much ATP is getting used by just this one cellular process. Okay, so now these things don't. So actin just doesn't spontaneously grow, though, right? Their growth is initiated by particular proteins, right? So, and it can be initiated in different arrangements. So these different complexes here can initiate the growth of actin. These formin proteins, they can initiate just a linear growth or a linear elongation at the plus end. So the formins will basically dimerize, and it acts as a region for the globular proteins to stick on. All right, other proteins, the ARP23 complex. So this causes like these branch networks, right? So this would just cause like actin to grow linear, right? So there's your formin dimer, and the trimer can stick on, and you can get growth at the plus end. But now if actin needs to branch, it can use other proteins to kind of come in and stick to the already created filament, and now actin can branch off. So this is ARP23, and these are the formin proteins. Actin also has caps on the ends that help give it strength. And also there's another protein called tropomyosin, which is involved in muscle contraction. Tropomyosin has like this long rope-like structure which wraps around the actin and gives it additional tensile strength. Okay, additional proteins can arrange the actin in these like parallel arrays, which we call bundles. And it can also arrange the actin into right angles. And we call these networks. So bundles and networks. There are also proteins that can sever actin, right? So if you break the actin molecule, now you're giving the molecule another opportunity to grow. Now because from one you've created two, and they can both grow. So you're going to get two longer molecules from just one. So it's the Coughlin proteins that break up actin. And interestingly, these proteins respond heavily to changes in calcium levels in the cell. So we're going to talk a little bit about calcium today when we do these data sets. Actually, so we'll just wait till then to do that. But calcium fluctuations can initiate or recruit these Coughlin or these actin severing proteins. All right, so on the left there, you can see the actin filaments and the cross-linking proteins that cause it to produce bundles, right? Which is just a series of parallel filaments. And then also the networks, which are produced by these proteins that cross-link to produce right angles. All right, so there's the minus and plus end of actin, the capping proteins, and also the Coughlin protein, how it's splitting the filament, and now you have the ability to grow two more filaments. So a really good model cell system to study actin is the erythrocyte. Why do you think that is? Why is the red blood cell really good model cell for studying cytoskeletal proteins? They have a unique shape, and that shape is pretty rigid, right? So they have, red blood cells are trying to maximize their surface area to maximize exposure to hemoglobin. They're also they need to maintain like some flexibility, right? So and we're going to talk about that, like what makes those erythrocytes flexible. Why is it important that red blood cells are flexible? So they can fit in capillaries, they can squeeze in there so they don't clot up as easily, they don't bundle up. So yeah, erythrocytes have a biconcave shape. So it's not like a donut, there's no hole in it, but it looks caves in on both sides. That increases the surface area. And this is all modulated by the arrangements of actin. So also red blood cells, have no nuclei. So, you know, um, their physiology is really directed at maintaining hemoglobin and maintaining a certain cellular shape. So good model system. All right. One of the main proteins that interacts with actin underneath the plasma membrane is this protein called spectrin, and we're going to see that. And it's spectrin that actually gives the cell its elasticity. So spectrin is a tetramer protein. There's 2 alpha and 2 beta chains. And then sticking out from the beta chains on the side are these actin binding domains. So actin is going to stick on both sides to spectrin. And spectrin has these alpha helical regions within it that make it very elastic. Right, there's the spectrin protein. You can see the 2 alpha chains and the 2 beta chains, and then the actin binding region, the binding domain that's on the ends of the beta chains. So that's where actin is. So this is, think of it like a spacer molecule. It's going to exist between actin filaments and give you some flexibility. So all of this would be lining inside the plasma membrane. All right, if you're looking at the cortical cytoskeleton in these erythrocytes, like I said, the spectrin tetramers are going to exist right under the plasma membrane, and they're going to be cross-linked to the short actin filaments at the actin binding domains. Right, it gives the cells their shape, and it has to be connected to the plasma membrane. You can't just have this thing loosely laying under the plasma membrane. There are anchors that like span the plasma membrane. They're transmembrane proteins that bind to actin and spectrin. Right. There is a protein that's related to spectrin that's found in skeletal muscle called dystrophin. And it's problems in this protein that contribute to all of the problems that people with muscular dystrophy have. All right, so here are the anchor proteins. You can see them spanning the membrane, and they're binding both to actin. So the glycophorin is binding to actin, and band 3 proteins bind to the spectrin. So, all of this complexity in terms of like all these different parts that make up the scaffold gives it its rigidity. It also allows it to form into certain shapes, and gives it its strength as well. Okay. All right, so the last part of this section looks at 2 additional components. So how does actin link to the outside world of the cell? Right. So when we're talking about that, think about it in 2 ways. So how does the cell bind to the extracellular matrix, right? The ECM. And that means how does it bind to proteins that are surrounding the cell? And then how does the cell bind to other cells? Okay, those are the 2 things that we're going to look at. So the first thing is, how does it bind to the extracellular matrix? So what is the extracellular matrix? Well, it's proteins that exist in the microenvironment that cells exist in. Collagen, for example, would be an example of an extracellular matrix. Collagen protein is all over our body. It's what sculpts our tissues, essentially. And the transmembrane proteins that exist in cells that bind to the ECM, that bind to collagen, are integrins, right? Integrins are the proteins that bind to collagen. If you're in, now it's not always collagen. If you're in the bone marrow, for example, there's a protein called fibronectin, which is very abundant. And integrins bind to fibronectin as well. These integrin proteins are not only like scaffolding and anchoring proteins. It's also been found that they're heavily involved in cell signaling. They serve as regulators for immune function, cell division, all sorts of things. All right. Typically, it's not, the integrins directly binding to actin. There's going to be adapter proteins as well. And that means the proteins that the integrins bind to and then eventually bind to actin. There's a few of them. Vinculin and talin are the most common ones. All right, here's our extracellular matrix. Imagine this is either fibronectin or collagen, and our integrin transmembrane proteins that bind to them that extend out from the cellular membrane. And here's our actin network inside. The yellow ones are the filaments. The cross-linking alpha actinin proteins kind of create that bundle orientation, right? And the talin and the vinculin are adapter proteins that connect to the cytosolic side of the transmembrane integrins and also bind to actin. Right, so complex network that provides anchoring to the extracellular matrix, but these molecules also provide signaling as well. A really good example of that, is in the bone marrow in terms of cancer treatment. So when cells are just kind of like blood cells, plasma cells are just really floating around the plasma, they're relatively more sensitive to chemotherapy than they are when they're bound in the fibronectin in the bone marrow. It's been readily shown that cells, when they are associating with the extracellular matrix, they're highly resistant to chemotherapy, and which with the chemotherapy. All right, so besides how the cell interacts with its extracellular environment, we also want to see how cells bind to other cells directly. All these cell-cell contact sites, also known as adherin junctions. Okay, and the main proteins that modulate these adherin junctions are cadherins. Okay? Cadherins are calcium dependent. So here is taking my cell culture class 1. Oh, you're not in it now. You're taking it next semester. Okay. Well, we didn't really do this in cell culture this semester because I couldn't get the cell line, because apparently it takes 6 months to order anything around here. But so when you have adherent cells, when you have cells stuck to the plastic dish, right? You have to get them off. You use a digestive enzyme called trypsin that breaks down the cadherins, right? But it's a very important step that you wash away all the residual serum and the growth medium from those cells before you treat with trypsin. And that wash solution, which is typically just saline, has to be free of calcium. People don't use calcium, it doesn't work. Cells don't come off. So cadherins are very calcium dependent in their binding. So if you're keeping calcium around, they're going to stay sticky. That's the main point. Anyways, you can see here you got 2 cells. This is a adherin junction, and these cadherin kind of protrude out, and they will connect. This is one cell, and this is another cell. They're like little fingers that are sticking out, grabbing each other. All right, there are adapter proteins for these cadherins as well, such as vinculin. That's a real common one. All right, tiny fingers, microvilli, or tiny projections. These are actual projections of the plasma membrane, but they're held in rigid form. Right? So plasma membrane extends out. Ultimately, what is that purpose does that serve? You extend out the plasma membrane. What are you doing? Why would you want to do that? surface area, right? The answer's right there. So, yeah, so the plasma membrane is coming out, but it's not just like sticking out there transiently. It's held as a rigid structure, and actin is the one that's producing it. It has additional cross-linking proteins like villian and fibrin that are doing it. And there's also some myosin proteins that help produce these microvilli structures as well. So you can see the bundles with the actin filaments going from the top of the microvillus to the bottom. It maintains its polarity. You have the plus and minus ends, and you can see the myosin proteins are attaching it to the plasma membrane, and also the cross-linking proteins that we talked about already, like vilin and fibrin. All right, these are related to the stereocilia, which are found in the inner ear. These are sensory cells that kind of are floating in suspension, and they are sensitive to vibration. Cell movement, The Blob. Has anyone seen The Blob before, that movie? I've never seen it. You've seen it?
I watched the very old,
You did? Was it good?
very silly.
It's silly, isn't it? Is it child appropriate? Is it kid appropriate?
I think the older one is, the newer one is.
Oh, there's a new one too?
Yeah, so like, they made one like.
I want to show that one to my kids. I might do that. You know what we did? There was a movie that came out. Was it Weapons? Anyone see that? That was awesome. The end. What an ending to that, man. That was pretty good. Anyways, Halloween, right? You guys should watch that movie. That's a good Halloween movie. All right. If you have kids, definitely watch it. Not with them though. Okay, so how does the blob move? Well, it moves basically in three different steps, right? So the end of it, like one side of it extends out. That's the first thing, extension. Second thing is attachment, right? So then that extension attaches to the surface, and then retraction. The back side of it caves in. That's how this thing moves. And the way it moves is through the use of like these false feet, right? These podia. So, or these different types of protrusions. So pseudopodia, lamellipodia, and filopodia, three different examples of different protrusions. Pseudopodia are important in phagocytosis, right? They'll wrap around. Lamellipodia are like flat, like think about like flat hands that can go out and stick to surfaces. And then filopodia are like little fingers that can go out and probe around the environment. All right, so some cool pictures here of that. Pseudopodia on the left and the middle. Right, and then this picture here, you can see where the L is. These are the lamellipodia, they're like flat extensions that can go out and strike surfaces and attach to surfaces. And then the filopodia is this thing right here. All right, the blob moving migration, like I said, extension, attachment, and retraction. Right, so extension is usually mediated by lamellipodia. Attachment is mediated by focal adhesions, which I don't know if I used that term before, but focal adhesions are the integrants, like how the integrants come together. And how does this all occur? Well, through branching and polymerization of actin, right? So it's rapidly, pushing the membrane out as it's growing. And then there are new actin-related proteins that come in and help branch it, like ARP23 that we talked about. Okay, there's the blob moving. All right, that's all I've got for you for this unit, which is unit 3. And then we'll have exam 3 on Monday. You guys have any questions about anything we went over today? Yes, ma'am. I know there's some. I hacked some stuff this morning,
Okay,
so
we're
I'll
not covering,
post another version of it today.
we're not covering row family protein and actin.
Nah, let's it out.
Mm.
This, what did I do? All right, you guys take a little break. But after this, we're gonna do some in-class data analysis. We're gonna do a little differently this time. So I'm gonna have you guys branch off. You're gonna work through some problems on your own, and then we're gonna, as a class, kind of go through them and discuss them. So go take a little break. I'll see you in 5-10 minutes.